Good morning, dear all. Today we are going to discuss about testis, which is male reproductive glands, which are paired and present extra-abdominally in the scrotal sac. So we can see in this diagram that these are testes, both side, right and left side, and this is the scrotal sac, and testis is suspended by spermatic cord of the corresponding side in the scrotal sac. Okay, so and this is scrotal ligament. Okay, that is another support for testis, and we can see this comma shaped structure that covers or overlaps the more or less full of the upper end and the posterior border and part of the lower end of the testis, that is known as epididymis. Okay, so in this picture you can see testis, epididymis, and this is spermatic cord. Okay, and this is scrotal sac. So they are extra abdominal in position, but during development in the fetal life, testis developed in the intra-abdominally, but later it descends and finally it reaches the scrotal sac. The most significant reason for this descent is spermatogenesis only occur at a lower temperature. Okay, so if it present within the abdomen due to any anomaly or failure of descent, then spermatogenesis will not occur and the person will be infertile totally. Okay, so this is the position of the testis in the scrotal sac. Okay, these are the main reproductive glands of male. Now, so you can see in the viscera that this is the viscera of testis. Okay, hope we can find similarity with the picture. So this is testis, this is epididymis. Okay, and this is the spermatic cord with the coverings that suspends the testis in the scrotal sac. You cannot see here scrotal sac as it is viscera. The scrotal sac lies like this. Okay, just like this. Okay, here scrotal ligament attaches with the scrotal sac on the base of the testis. Now coming to the detail of this viscera. So you can see this is more or less ellipsoid in nature. Okay, in shape, ellipsoid. Shape is ellipsoid and it has one upper end, one lower end. Okay, this is upper end. You can see one sessile pedunculated body in the upper end, that is known as appendix of testis. Okay, that is the developmental remnant of degenerated cephalic portion of the Mullerian duct. Okay, this is appendix of testis. You can see clearly from here that is present in the upper end of the testis. This is lower end. Okay, this area is lower end. So upper end, lower end. Now this is anterior border. We can see from, we are seeing from the side. This is anterior border. So you can determine that anterior border is smooth and convex. That is a curvature in front, is convex inside. And posterior border we cannot see properly from here. It is overlapped by the epididymis, head and body of the epididymis. So it is broad and flat. Anterior border convex and smooth, posterior border broad and flat. So there is upper end, lower end, anterior border, posterior border, and there is one lateral surface, another is medial surface. So how to determine lateral and medial and how to determine the site. Okay, this is very important. So if we hold testis like this, so this is the anterior border, this is the upper end, you can see. So how to determine lateral or medial end? You can see a groove here in between the testis and epididymis. only in the lateral surface, posterior part of the lateral surface of the testis. There will be such groove that is known as sinus of epididymis. So by seeing the sinus of epididymis, you can say this is the lateral surface of the testis, okay, and this one is the medial surface of the testis. So by this way you can determine the site, okay. So we can say from this specimen that this is testis of the right side because this lateral surface facing towards right, okay. So this is the right-sided testis, okay. Now coming to the different part. So this upper part, you can see this pedunculated body that is already told that this is appendix of testis. Lower part, it is connected with epididymis, okay. You cannot see separately. Now this lateral surfaces, medial surface, both are smooth and convex, okay. In case of lateral surface, in the posterior part of the lateral surface, you can see this groove that is sinus of epididymis, okay. Now coming to epididymis proper. So you can see this comma-shaped body. This is known as epididymis. This part that's overlapping the upper end of the testis, that is known as the head of the epididymis. This is body of the epididymis that overlaps the posterior border of the testis, and this part which overlaps a part of the lower end of the testis, that is known as tail of epididymis. So there are three parts: head of the epididymis, body of the epididymis, and tail of the epididymis, okay. From this picture you can see I already drawn here. So this is head of the epididymis, body of the epididymis, and tail of the epididymis, okay. Now another thing, now going to the coverings of the testis. The most important part in covering is in the front of the testis. So I am seeing a sagittal section here, the front of the testis, this spermatic cord, this is testis sagittal section, this is epididymis, okay. So what is this orange colored portion? This is known as tunica vaginalis, that is a space, that is the detached part of the processus vaginalis, okay. While testis is just see the another picture here. During intrauterine life, testis is in the developed in the posterior abdominal wall, okay, this picture. Then it descends gradually, okay, by the pull of this gubernaculum testis, okay. Just a rope or tie rope like structure, that is gubernaculum testis, is descends, okay, and then ultimately placed in the scrotal sac. This is the peritoneum, peritoneal sac, this portion. So while during descent, it takes a part of the peritoneum with it. You can see in the second picture, takes a part of peritoneum with it, and finally this part of the peritoneum gets detached from the total whole peritoneal cavity, and this part remains as tunica vaginalis in front of the testis as a covering, as a protective covering. It is remains as tunica vaginalis. This part is known as processus vaginalis, okay. The part of the peritoneum that were descending with the testis, and this detached part is known as tunica vaginalis. And you can see that there are two layers here. One is just closely approximated with the viscera testis, that is known as visceral layer of tunica vaginalis, and parietal layer of tunica vaginalis. So there is a covering in front of the testis. So now what are the other coverings of the testis? Testis from inside, from outside inwards, it is covered by, most outside there is presence of tunica vaginalis like this. Two layers are there, parietal layer of tunica vaginalis, then visceral layer of tunica vaginalis. Then there is another layer is known as tunica albuginea. Tunica albuginea, that is one fibroareolar membrane that is white in structure. Now this tunica albuginea covers the whole of the testis, but in case of visceral layer of processus vaginalis, it does not cover the whole testis. It covers the anterior border, medial lateral surface of the testis, but does not cover the posterior border where the epididymis is attached. The visceral layer of the tunica vaginalis does not cover that part. Okay. Next to tunica vaginalis, the next covering inwards, that is tunica albuginea, that is tough fibrous membrane. It covers the entire testis, and from the posterior part of the posterior border of the testis is going inwards as a longitudinal septum, incomplete longitudinal septum known as mediastinum testis. Okay, from this posterior border it goes inwards in the substance of the testis, and it forms a incomplete longitudinal septum that is known as mediastinum testis. From this mediastinum testis, from the convex border of this mediastinum testis, so like this mediastinum testis, from the convex border of the mediastinum testis, numerous fibrous septa appears and goes again anteriorly and divides the inner mass of the testicular tissue into different lobules, numerous lobules, and in each testis there is approximately 200 to 300 lobules are present. Now the innermost covering of the testis is tunica vasculosa. It is formed by the loose areolar tissue, and it covers the each and individual lobule, okay, and the blood vessels related with it. So there are mainly three coverings of testis. Most outside there is tunica, visceral layer of tunica vaginalis, then next to it tunica albuginea, that forms the mediastinum testis, going inside of the testis and form the mediastinum testis, and another is tunica vascularis. So these are the coverings of the testis. So here we can see one section of testis, but you cannot determine from here what is the exact inner structure. So this each lobule, I have said already that septa, fibrous septa from mediastinum testis divides the testicular tissue into numerous lobules. Okay, so each lobule contains 1 to 3 seminiferous tubules. Okay, that is the tubule where the sperm is being produced. Okay, now seminiferous tubules, the seminiferous tubules has convoluted part in front and straight part behind. Now see from this diagram that this is the interior of the testis. Okay, so we can see that this is the anterior part, so this is the posterior part. Now seminiferous tubule, there is convoluted part, means coiled part in front and straight part behind. Okay, these are the straight part of seminiferous tubule. So this part is mediastinum testis. Okay, that is formed by the inward projection of the tunica albuginea. In the mediastinum testis, this straight part of the seminiferous tubule joins with each other and form reti testis. Okay, that here the ducts is known as reti testis, and this reti testis joins together in the head of the epididymis, where it forms the efferent ductules. Okay, coiled efferent ductules. It forms the efferent ductules in the head of the epididymis. Then these efferent ductules forms the canal of epididymis in the body and tail of the epididymis. Okay, so from this seminiferous tubules that is convoluted in front, straight behind, the straight part of seminiferous tubules joins with one another with the adjoining seminiferous tubules and forms the retitestis here, and then this retitestis united to form coiled efferent ductules in the head of the epididymis. Then this efferent ductus forms the canal of epididymis that traverses the body and tail of the epididymis, and finally it forms vas deferens. Okay, let's see this picture. So you can see testis here. So this is epididymis. So from here the vas deferens begins. From the tail of the epididymis, vas deferens begins. This violet color structure, this tube is vas deferens, goes upwards through the superficial inguinal ring, then inguinal canal, then deep inguinal ring, then finally it drains into the, this is prostatic urethra, this is prostate gland, this drains into the prostatic urethra after joining with the seminal vesicle. Okay, this is vas deferens, is coming from the tail of the epididymis, goes like this from superficial inguinal ring, deep inguinal ring, then ultimately drains in the prostatic part of the urethra with seminal vesicle. Okay, this is the pathway of sperms. Okay, so about 12 to 20 efferent ductules appear from the upper end of the epididymis. Well, how efferent ductules are being formed? So here within the testis there are lobules, at least 200 to 300 lobules in each testis. In the lobules, in each lobule there are 1 to 3 seminiferous tubules. In the seminiferous tubules, the anterior part of the seminiferous tubule is convoluted and the posterior part, that is towards the epididymis, is rather straight. Okay, anterior part convoluted, posterior part is straight. Now the straight part of the seminiferous tubules joins with one another and form retitestis in the mediastinal testis. Okay, you can remember mediastinal testis. So in the mediastinal testis it forms retitestis. Now this retitestis joins and form the efferent ductules, here coiled, and form the efferent ductules in the head of the epididymis. Okay, then again the efferent ductules convoluted and forms body and tail of the epididymis. Okay, and the canal of epididymis. Okay, now this canal of epididymis is continuous with vas deferens, which is present, which is started here in the medial part of the posterior border. Okay, so it is continuous with the vas deferens. Now coming to the arterial supply. Okay, arterial supply is mainly by testicular artery, that is a direct branch of abdominal aorta. Okay, so other supply that is present for testis is from cremasteric vessels, that is this branch from the inferior epigastric vessels, and also from the artery to the vas. Okay, artery to the vas, that is branch of the superior or inferior vesicle artery. So there are three sources testis is being supplied. One is testicular artery, mostly it is supplied by testicular artery, most part, that is the direct branch of the abdominal aorta. Another is artery to the vas deferens, that also gives a tweak to the testis. Artery to the vas deferens is a branch of superior or inferior vesical artery, and also from the cremasteric vessels, that is a branch of inferior epigastric vessels. Now coming to the venous drainage. Venous drainage is important for testis. The posterior border of the testis, approximately 15 to 20 veins are present that unite to form pampiniform plexus, that is known as pampiniform plexus of veins, and this forms the main bulk of the spermatic cord. Pampiniform plexus of veins form the main bulk of the spermatic cord, and it helps in the regulation, that is thermoregulation of testis, because spermatogenesis is only possible, I have already told, spermatogenesis is only possible in the lower temperature, that is outside the abdomen, that is not possible inside the abdomen. That's why testis descended in the scrotal sac. So this pampiniform plexus helps in maintaining the thermoregulation, means it keeps the testis in a favorable, it maintains a favorable temperature for testis, so that the spermatogenesis is uninterrupted. Okay, so this 15 to 20 veins from the posterior border of the testis unite to form pampiniform plexus, which forms the main bulk of the spermatic cord, and at the superficial inguinal ring. So in the superficial inguinal ring, the veins joins to form 4 veins, and the deep inguinal ring, the veins joins to form 2 veins. Finally, the 2 veins again join to form 1 vein, and in the right side, the vein drains directly in the inferior vena cava in an acute angle, and in the left side, the vein drains into the left renal vein almost in right angle with the left renal vein. Okay. So you can see from this picture, this is testis. So these are the pampiniform plexus of veins. At least 15 to 20 arise from the posterior border of the testis, unite at the superficial inguinal ring. Then 4 vein traverses in the inguinal canal. Then in the deep inguinal ring, it is only 2 veins are there. Then again this unite, and the posterior abdominal wall, it drains into the inferior vena cava. In case of right side, drains directly into the inferior vena cava. In case of left side, it drains into the left renal vein. The angle is almost 90 degree, and here it is an acute angle. Okay. And another thing is need to be mentioned, that is the left testis lies at approximately 1 centimeter lower than the right testis in the scrotal sac. Okay. Now coming to the lymphatic drainage. Lymphatic drainage of testis is mainly in the pre and para-aortic lymph node, and nerve supply, it is mostly supplied by sympathetic nerves from the aortic and renal plexuses. Preganglionic fiber derives mostly from the 10th and 11th thoracic segments of the spinal cord. That's why testicular pain sometimes can be felt around umbilicus. So that's all. Thank you.